Hi guys, and here we are with video number 2. Now we're going to move on to introducing the mathematical model in general. So first, think for a minute about what a model is. Think about all the different ways that we use models. Not just mathematical models, not just in biology, but in general, all the ways that we use the term model. Here's a few hints to help you. First of all, it could be a map that helps us model geographical space. It could be an organism that helps us model other organisms. It could be an equation or some other sort of math that helps us model a process. It could be a model building or train or car that helps us model a thing. It could be a diagram like this that helps us model a concept or a bunch of concepts or a process. So what do we mean when we use the term model? So what I think of when I use the term model is that it's a simplified representation of an object or a process. So this isn't everything about geographical space, it's just the most important things. Drosophila obviously doesn't have everything humans have, but it has enough in common that it's useful. It's just simpler. Same for all of these things. It's a simplified representation of an object or a process. And in order to simplify, we have to make some assumptions about what's important and what's not important, what can be simpler. So I really like this quote. Essentially, all models are wrong because they're not completely reality, but some are useful. Because honestly, it would be impossible to do anything with complete reality. It's so complicated. So you have to determine which assumptions are worth making, which assumptions are helpful, and which assumptions are going to completely obscure the kind of process that you're interested in. Okay. So what mathematical population models do we use? Well, you've all heard of exponential growth, where the rate of population growth is directly proportional to the per capita population growth is a constant. So population growth is directly proportional to the number of individuals in the population. So as the population increases, the population goes faster and faster and faster. It accelerates. We've also talked about logistic growth, where the population initially grows exponentially, but then slows down and kind of levels off at this carrying capacity K. And that's what the equation looks like. So when we're trying to determine which mathematical model to use to model a particular population or process, we have to go through a bunch of different steps, and then also to analyze it and actually figure out what's going on as well. So first you want to figure out exactly what your research question or questions are, I guess. Then you want to determine which assumptions you need to make that won't obscure the answer to your research question. And then you have to figure out what your conceptual model is, which is usually some kind of drawing or diagram. And then you figure out what the form of your mathematical model should be. Whether it's a matrix or an equation, or whether it's discrete with time steps or continuous that's constantly changing. You have to assign values to your parameters. So what are parameters? They're basically those constants that you see in equations. So for instance, for logistic growth at R, for logistic growth both R and the K are parameters. So they're values that are used in the mathematical model that the modeler decides on when they run the model. And then finally, we can use the model to actually answer our research questions. So now we'll go on to talk about structured population models, also known as matrix population models. So first you have to ask your research questions. So for population models, we're usually asking, is the population growing? And once you get into more of the structured population models, you want to ask what stage or what demographic processes should managers focus their monitoring and conservation effort on? How might different threats or management actions impact the population? And then you have to determine what assumptions you want to make. So for exponential growth, we usually assume that the environment is constant, it's not changing. Alternatively, the environment could be stochastic, in which case it's randomly changing. We usually assume that the environment provides unlimited resources, so you can have constant birth and death rates. So the birth and death rates don't depend on what the population size is, or the population density. If that's not true, then you might end up getting logistic growth, depending on exactly how you model it. For both exponential and logistic growth, we generally assume that there's no immigration or emigration, no individuals moving in or out of the population. We assume that there's no time lag, so the population growth rate reacts immediately to how many individuals there are in the population, or at most in the last time step, not multiple time steps ago, which would be a time lag. And we assume that all individuals are identical. So what kind of organisms do all these assumptions actually make sense for? Think about that one for a sec. Yeah, I'd say it pretty much only makes sense for bacteria. It's very unlikely that resources are going to be unlimited and that all individuals are identical. So if you change this assumption about unlimited resources, then you end up getting logistic growth. And if you change this assumption that all individuals are identical, then you can look at populations that have complicated life histories, like, honestly, most organisms. So today we'll be using examples of Laysan albatrosses and also lionfishes. So again, if you get rid of that assumption that all individuals are identical, you can instead say that all individuals in each stage are identical. And then you have to change this birth and death rates to vital rates, which is basically the rate of transitioning from one stage to another. So again, the big difference between the assumptions for exponential growth, which are in black, and the assumptions for a complex life history for structured population model, which is in red, is that instead of all individuals being identical, you instead assume that all individuals in a particular stage are identical. So again, this gives you the structured population model or matrix population model. So structured population models can be structured in a bunch of different ways. So basically, you have to determine how your population can be divided up in a way that makes the most sense. So it could be that it's structured by age, size, sex, or developmental stage. So in the albatross population we'll be talking mostly about age, because albatrosses mostly do things at certain ages. But in other organisms, like fishes, for example, it makes a lot of sense to look at size. In some organisms, like say the pearl oyster's eye study, they can change sexes, so looking at the different sexes actually matters. In most population models, we assume there's plenty of males around, and that the population is actually limited by females, so we actually only usually model the females. It could also be the developmental stage. So, for example, for the lionfish population we'll be talking about different developmental stages. This could also be true for, say, insects that have their larval stage. And once you've decided how to divide the population into different stages, whether by age, size, sex, or developmental stage, you have to put it all into mathematical terms. So we have this vector that's the number of individuals in each stage at time t plus 1. We have this matrix that's the probability of transitioning from one stage to another for all the stages. And so you have both the number of rows that matches the number of stages and the number of columns, the number of rows that matches the number of stages and the number of columns that matches the number of stages. And you multiply this matrix by the population vector times t in order to get the population vector at time t plus 1. So what do all these numbers mean? So again, the two population vectors, n t plus 1 and t, is the number of individuals in each stage at a particular time, which is usually determined at time 0 by the modeler, and then after that by multiplying by this matrix. So it's determined by the model after the first time. Okay, so for most organisms that can only grow bigger or reproduce, this is what it will look like. Organisms can stay in the same stage, which is what we'll call persistence, or P. So here, this is the probability of an individual in stage 1 of staying in stage 1. Here, it's the probability of an individual in stage 4 staying in stage 4, persisting in stage 4. So that's our main diagonal here. Just below the main diagonal is growth. It's the probability, for example here, in column 1, row 2, the probability of an individual that's previously in stage 1 of growing into stage 2. Or down here, it's the probability of an individual previously in stage 5 growing into stage 6. So if you have an age-based model, basically individuals are only growing. They can't stay in a stage for more than 1 year. And then there's also fecundity. So, for example, F4 is the number of individuals that one individual, usually female, in stage 4 contributes to stage 1. So basically each of these is the contribution of the stage, that's the column, to another stage, that's the row. Either probability or the number of individuals reproduced. For a population in which individuals can only reproduce, or age, or grow larger, or go to the next stage, all the rest of them are going to be zeros. For more complicated life histories, where individuals can get smaller, or skip a stage, or more complicated for whatever reason, you might not have the rest of those all be zeros. There might be non-zero values here or here as well. But today, we'll be focusing on one that isn't quite that complicated. Okay, so let's start with a really simple example. There's only 3 different stages of lionfish, where there's larvae, juveniles, and adults. And in this case, because individuals have a pelagic larval duration, are larvae for about a month, we use a time step of a month. So here, we're going to put what we know about the life history onto a conceptual model, in this case, a life cycle diagram. So L for larvae, J for juveniles, and A for adults. So first, what stages can grow from one stage into another stage? Well, larvae can grow into juveniles, and juveniles can grow into adults. So we'll label those with G's. What stages can persist for more than one time period, which is, in this case, a month. The larval duration is only about a month, so they can't persist. But you can be a juvenile or an adult for more than one time step. So those have p-values that are not 0. And what stages can reproduce? Well, pretty much by definition. Adult means able to reproduce. So only